Hello, everybody, and welcome to the MycoTalks. This is the first MycoTalk of 2022. My name is Ilya Jacobsen, and I'm at Weill Cornell Medicine here in New York. And my co-chair today is Dr. Gordon Brown from Exeter University. We have two exciting talks for you from Dr. Ilse Jacobsen and Dr. Alistair Brown. For those of you that are new to the MycoTalks, I'm sorry that you missed season 1, but there is a good news. There is a season 2 right now, and a lot of exciting talks that are coming up. So the philosophy of the MycoTalks is that we have a diverse audience in terms of and presenters in terms of career stage, gender, geography, and field. The MycoTalk, each talk is 30 minutes. With a Q&A session at the end. So please post your questions in the Q&A on the bottom of your screen, and we will moderate your questions. There is a recording as well. So for those of you that miss the talks, they can go on the YouTube channel and re-see them. We have two upcoming talks that I would want to tell you about in February and in March. So please register for those sessions. A lot of exciting talks coming up. For trainees, there is a medical mycology trainee seminar series, which are focusing on students, postdocs, clinical trainees, and new faculty. Please register, watch them, and please submit also a proposal for your talks. You might be selected as a speaker. It's exciting. And I will finish my intro here with an exciting announcement of a movie premiere, a film by Leonie Hampton, Our Body is a Planet. So that will feature a lot of our small friends, fungal friends here, and the premiere will be at the Exeter Phoenix and online on the 3rd of February. So please register. There is a link for those of you that watch, for example, Fantastic Fungi on Netflix. I'm sure you will like this one very much. So without further ado, I will move on here. And present our first speaker. So Dr. Jacobsen is a professor at mucosal immunology at Friedrich Schiller University in Vienna, and is the head of the laboratory of microbial immunology at Leipzig Institute for Natural Products. So she received multiple awards for her contribution to the field, the MEDIAC Research Award and several publication awards. Her group focuses on the interaction between Candida and the host, aiming at better understanding of the interactions that lead to pathophysiological alteration, and strive to understand which processes in the pathogenesis lead to the development of clinical symptoms and lethal disease. So they are focusing on several main areas: the role of fungal morphology in pathogenesis, molecular mechanisms contributing to Candida sepsis, and gastrointestinal colonization and translocation. So the seminar title is The Role of Filamentation in Systemic Candidiasis. Ilse, we are looking forward to your exciting talk.
Thank you very much, Ilya, for this very kind introduction. And I would like to start with saying that I'm really very grateful for having been given the opportunity to present some of our work here today, and that I love this seminar series. I think it has been a great success, now starting with season 2, and I hope it will run into season 10, 11, and 12 in the future. So systemic candidiasis and the role of filamentation in this disease is actually a topic where, when I entered the field a bit more than a decade ago, I would have thought is well done. You know, we know that filamentation is essential, and the question would just be how many more genes are involved in driving filamentation. However, over the last couple of years, we've been working with a mutant with an interesting and counterintuitive phenotype. We recently published that work, and I will summarize it in the first part of my talk. And then I'll try to link it to some previous work where actually we talk a little bit about farnesyl as a quorum sensing molecule. And finally, in the end, I will present you some preliminary data of ongoing work connecting the two. As most of you will know, Candida albicans is an opportunistic fungal pathogen and one of the few species within the genus Candida that's truly polymorphic, meaning that it grows as typical spherical yeast as well as pseudohyphae, and it's also able to form true elongated hyphae. Yeast to hyphae transition has been used for decades as an important diagnostic feature, but since 20-30 years, we know that it's also a key virulence factor. In fact, if you look into any of the different forms of candidiasis Candida albicans can cause, you will always find hyphae as a hallmark of fungal invasion, both in mucosal forms of candidiasis, such as oropharyngeal infections or vulvovaginal candidiasis, but also you will find hyphae in various internal organs that can be affected by deep-seated or disseminated or systemic candidiasis. So how do hyphae contribute to pathogenesis? Well, one explanation is relatively straightforward. If you look at what hyphae do, and you know about the immense turgor pressure these cells can build up, it's easy to imagine that they're able to physically pierce through cells, as you can see here, as well as tissue. As you can see on the left-hand side, this actually is a murine liver after peritoneal infection, and these pink structures invading deep into the organ are all Candida albicans hyphae. Hyphae are also involved in immune escape because they're one of the factors allowing Candida albicans to escape from phagocytes, especially macrophages. And besides these physical properties, the transition from yeast to hyphae is also associated with significant changes in transcription and the expression of so-called hyphae-associated genes. And amongst those are several well-characterized virulence factors. Maybe the most important and most recent addition to the list of hyphae-associated genes in terms of virulence is EC1, which encodes the peptide toxin Candida lysin. Another aspect, when you think about yeast on the one side and hyphae on the other, is that the morphological transition leads to changes in cell wall architecture. And this in turn might affect the exposure of pumps and interaction with immune cells. And I assume this is a topic Al will talk about a bit more in his talk. Maybe the most important piece of evidence for the very important role filamentation plays for Candida albicans infections comes from murine models of disease. So a very commonly used model is intravenous infection. This is resembling systemic candidiasis and catheter-associated infections in humans. And more than 20 years ago, it was shown in the Cell paper by Lowe and co-workers that EFG1 CPH1 double mutant, which is unable to form filaments in vivo, is at the given infectious dose avirulent in mice. Stephen Saville, a few years later, expanded on that, looking at NRG1 as a repressor of filamentation, and could show that if NRG1 is constitutively expressed and the fungal cells grow in yeast only, the strain is avirulent and virulence can be switched on and off by regulating NRG1 expression. Another mouse model of candidiasis is abdominal candidiasis induced by intraperitoneal infection. And also in this setting, filamentation is important for organ invasion, organ damage, and clinical disease. So what I will talk about today all started many years back when I worked as a postdoc with Bernhard Hube. And Bernie's lab had identified an interesting gene where the deletion mutant showed a peculiar phenotype. The mutant is still able to form short germ tubes, but just after germ tube induction begins switching back to yeast cell growth. So it's unable to maintain filamentation. And if you look at the culture, after 12 hours, you will see yeast cells only. In an infection model using reconstituted human epithelium, so a multicellular layer, this results in a situation where the mutant is able to adhere and invade, but then switches back to yeast growth and is trapped in the superficial cell layer. And this then consequently leads to less damage induction compared to the filamentous wild type. Now, this is not only the case for oral epithelial cells, but you also see a strong reduced ability to damage host cells when you use hepatic or renal cells in cell culture. Now, we tested the deletion mutant in an intraperitoneal infection model, and as expected for non-filamentous strain, we got less clinical symptoms in mice infected with the mutant. When we looked at markers for liver damage, here ALT, or pancreatic damage with the pancreatic amylase, there was much less released for the mutant in orange than for the wild type. And this was not due to an inability of the mutant to thrive in vivo, because the fungal burden in the liver was actually significantly higher, over a log higher for the mutant than the wild type. So this was consistent with what we expected. Lack of filamentation resulted in strongly reduced virulence. Just to complete the picture of the mutant, we decided to also do an intravenous infection model. And there we were in for a surprise. So at our standard dose we usually used, we saw only a very minor difference between the deletion mutant and the wild type. And actually, the mutant seemed to be a little bit more virulent than the wild type, the complete opposite of what we expected. And to be honest, when we got this data back, the first thing we did is re-isolate colonies and check to make sure that we hadn't switched accidentally strains. But it really was the ED1 mutant. When years later we repeated those experiments, we got the same results, and with a lower infectious dose, where all but one mouse infected with the wild type survived, the mutant was still able to induce lethal infection in more than half of the animals. So contrary to what we expected, we didn't see any attenuation of virulence of the ED1 mutant in systemic bloodstream infection in the mouse model. Now, before we went into trying to figure out why that is the case, we really wanted to be sure that what we were looking at was not an artifact, any kind of pleiotropic change due to the mutagenesis procedures. So we decided to construct a second set of strains based on the work of Stephen Saville with NRG1 using a TET-off system. So in this system, without doxycycline, the mutant strain is able to form filaments, and it's as virulent as the corresponding wild type in the absence of doxycycline. However, if you add doxycycline either via the drinking water or the mouse food, filamentation will be repressed by reduced transcription of ED1. So these two images here are micrographs of kidney sections. In pink, you see fungal elements, clearly hyphy formation for the wild type in the presence of doxycycline, whereas the ED1 mutant with doxycycline grows exclusively, more or less, as yeast cells. Despite this clear difference in morphology, survival of the mice was identical in both infection groups. So why do these mice die after infection with the ED1 mutant with doxycycline in the absence of Hyphy? The first hypothesis we tested was that maybe there is transcriptional dysregulation in the mutant, and the yeast cells of the ED1 mutant produce Hyphy-associated genes, but this was not the case. Another hypothesis or explanation could be that even though clinical disease and mortality looked identical, pathogenesis was different. So systemic candidiasis leads to organ damage by two different mechanisms. The first is direct fungal growth, invasion into tissue, and tissue destruction due to growth and expression of cytotoxic virulence factors such as Candida lysine. Now, due to the absence of Hyphy for the mutant and the lack of expression of Hyphy-associated genes, we would expect that the direct damage caused by the fungus would be reduced. However, this might have been compensated by increased immunopathology, so collateral tissue damage driven by recruited pro-inflammatory immune cells, especially neutrophils. To test this hypothesis, we performed time course experiments. and did extensive immunoprofiling. I'll just show you snapshots of the data and summarize the main findings. So if we looked at the total number of CD4 positive cells in the kidneys of infected mice, so immune cells, we found that up to 48 hours, there was literally no difference between wild type and ED1 infected kidneys. There were significantly more immune cells after 72 hours, but after 72 hours only. We also looked at immune cell subpopulations, and this is the example for 24 hours after infection. And there we saw for early time points, absolutely no difference between wild type and mutant. The different subsets were recruited in similar numbers. And after 72 hours, we saw some differences, which were also reflected in total leukocyte numbers. When we looked at cytokines, and we used a multiplex panel to look at a large variety of cytokines simultaneously in single samples, we were a little bit surprised that at 24 hours, when we saw the same number of leukocytes being recruited and the same subtypes, we found less pro-inflammatory cytokines in the kidneys infected with the mutant versus the wild type. Now, this changed over time. There was much less difference after 48 hours, and after 72 hours, as well as in moribund animals, there was increased production of pro-inflammatory cytokines after infection with the mutant. So there are differences in the immunological response. In immune cell recruitment, there are more immune cells recruited with the mutant at late time points and late time points only. The cytokines are strongly time dependent with less pro-inflammatory cytokines early on, but more at late time points. We did, however, not observe any differences in markers for systemic inflammation, so sepsis markers. And when we measured kidney damage by molecular renal damage markers and markers for kidney function, we also found no difference. So this suggests that there might be enhanced immunopathology. We have some indications, especially for the late time points. But we wanted to really test this further, and therefore decided to simply take away the cells that mediate immunopathology in mice, so neutrophils and pro-inflammatory monocytes. If you deplete these cells with a monoclonal antibody, the mice become highly susceptible to systemic candidiasis. So this is a really low dose. For a 20-gram mouse, that's 2 × 10⁴ cells in total per mouse. And as you can see, if you use the wild type Candida albicans, this leads to full mortality within less than 24 hours. With the ED1 mutant, clinical disease is delayed. The onset of symptoms is later, and then it progresses as quickly as with the wild type, but this difference is significant. So if you remove the opportunity of immunopathology, you make the ED1 mutant less virulent than the wild type. And contrary, if you take immunocompetent animals and the same infectious dose, the mutant is more virulent than the wild type. When you look in the immunocompetent animals, histologically, for the wild type, for those mice which survived, it's really hard to find fungal cells in the end, and there's only a few immune cells scattered around in the tissue, whereas for the ED1 mutant, you find groups of fungal cells and large infiltrates with immune cells. So suggesting that really this increased virulence in immunocompetent mice is driven by immunopathology to some extent. However, this difference in the immunological responses does not fully explain the virulence, and there's always a second side to stories, and in this case, the second side is fungal burden. So within the time course experiment, we also quantified fungal burden and found striking differences between the strains. On the one side, we have, this is data with the doxycycline regulable strain. In red, the ED1 mutant with doxycycline, so growing in the yeast form only. And then we have the 2 wild type controls plus minus doxycycline, and ED1 without doxycycline being able to form filaments. So across the filamentous strains, you see similar fungal burdens, but the red line sticks out those are the yeast cells with significantly higher fungal burden in the kidneys as early as 24 hours after infection, starting from the very same number 6 hours after infection. And similarly, we found increased fungal burden for the ED1 mutant in liver, brain, and spleen, although there the difference were more pronounced at later time points. Now, for those of you working with Candida albicans, you know that comparing yeast and hyphy numbers by CFU might be prone to technical errors due to the clumping or possible clumping of hyphy and the fact that you actually have filaments consisting of several segments with a higher mass, dry weight than a yeast cell would have. So we additionally performed DNA extraction from samples and quantitative PCR. And the data is what you can see here. So across a range of CFUs, and these are the very same samples depicted here. And we got a reasonably good correlation of the CT value and the CFU, independent of the morphology. So this cannot be explained by technical issues. So importantly, starting at 24 hours after infection, we have a much higher fungal burden, especially in the kidney with the ED1 mutant. And if you look at moribund animals, this difference is approximately 150-fold, so quite substantial. The 24-hour time point might be important because remember, I told you that we see increased leukocyte infiltration in the kidney at late time points, and this might be a consequence of the higher fungal burden, resulting in stronger stimulation for immune cell recruitment and also production of pro-inflammatory cytokines. Now, one question for the fungal burden data for us was, is this increased proliferation of the strain in the kidney and the other organs, or is it a result of reduced clearance by immune cells? And one way to answer this question is to turn back to our immunosuppressed mice, which lack the main effector cells able to kill Candida albicans, namely neutrophils. And here you see that in the kidney, but also other organs, starting at early time points when all the mice are still clinically healthy and alive, we already see a significant difference. This is increasing over time. And the biggest one we see in moribund animals. However, I'd like to point out that we're looking at different time points here. So histologically, we confirmed that we have yeast only in the ED1 infected mice in this setting, and that we don't have infiltrating immune cells. So in addition to hematology, we confirmed in the tissue that these mice really lack infiltrating immune cells. It also means because there are no immune cells, it's unlikely that pathogenesis is driven by immunopathology. So the lethal infection in these mice infected with the ED1 mutant must somehow be driven by yeast cells. So to briefly wrap up, for the ED1 mutants, we saw, consistent with everything published in the literature, that you do need hyphy for deep invasion, substantial tissue damage, and clinical disease in the murine abdominal candidiasis, the IP infection model. However, in systemic candidiasis, the ED1 deficient yeast cells are still able to cause lethal infection, and this coincides with a significantly higher fungal burden in kidneys and other organs. So how could higher fungal burden lead to lethal disease in the absence of filamentation? Well, the kidney is an organ with a relatively tough capsule, which cannot expand if something is going on in the organ and increasing, for example, the number of cells, like the large number of yeast cells. So this might lead to increased pressure, which in turn could damage host cells and lead to organ dysfunction. Another possibility is simply that yeast cells do have virulence factors, which are just much weaker than what we find in hyphy, but that the larger number of yeast cells compensates for the lower damage relatively per fungal cell. It's pretty hard to dissect to which extent one of these potential mechanisms does in fact contribute to tissue damage in vivo. If anybody has an idea, let me know in the discussion. Good. So take-home message here is that significantly increased proliferation of yeast cells in vivo can compensate for the lack of filamentation in Candida albicans. Now, proliferation points towards metabolism. So we did the obvious thing. We performed growth curves of wild type and mutant on different carbon sources at 37 degrees. And what you can see here is that depending what media you use, at low glucose as a sole carbon source, the mutant has a disadvantage. However, if you use alternative carbon sources which are present in vivo, such as glucnac or citrate, as well as BSA, you get better growth with the mutant compared to the wild type. And this also is the case if you do kidney homogenates, filtrate them, and use them as a growth media. We did do transcriptional profiling, hoping that we might find any pathways differentially regulated, which could explain these differences in growth, but we unfortunately were not able to identify any distinct pathway. So how could then ED1 be mediating this growth advantage under in vivo conditions? The predicted protein encoded by the ED1 gene is relatively long, close to 900 amino acids, and you can differentiate 3 regions. The N-terminus, which has a 36 amino acids called col domain, then a glutamine and proline-rich central region, and a C-terminus, which lacks any kind of specific domains or other obvious features. So based on this, we do know that ED1 is not a classical transcription factor. It might be interacting with other proteins. However, we were not able to really follow this further. And this might have to do with the actual protein. So the in silico prediction is that the ED1 protein is overall unordered, of low complexity and high instability. And the high instability might explain why we were unable with several approaches and different collaboration partners to heterologously express the ED1 protein or to detect the native ED1 protein with antibodies directed either against peptides from the C-terminus or the N-terminus. So we were unable to get our hands on ED1 as a protein and to test for potential interaction partners. However, we have some idea of what ED1 deficiency is doing besides enhancing growth in infection models. And this is a second older publication I want to briefly mention. So Melanie, when she started as a PhD student in my lab, as some of the first experiments she did was just to get familiar with the culture of Candida albicans, and she used filament induction on a plastic surface in RPMI 37 degrees and high CO2. And she found that the germ tube induction, which is what the ED1 mutant can still do, was strongly affected in the ED1 mutant by the density of the inoculum she used. This pointed towards chrome sensing and farnesyl. And indeed, the ED1 mutant turned out to be highly susceptible to the germ tube inhibiting or filament inhibiting effect of farnesyl at very, very low concentrations, where we see no effect on the wild type whatsoever. So at that point, we decided to use this hypersensitivity to farnesyl as a way to maybe tweeze out which pathways ED1 is involved in by targeting pathways which are known to respond to farnesyl or to be involved in the response to farnesyl. So Melanie constructed single and double deletion mutants in wild type and ED1 deficient backgrounds, targeting the different factors here marked in red. So we don't need to go through the whole detail of the complex signaling cascades affected by farnesyl. A lot of work went into this, and we are confident that we can exclude ED1 being involved in CSF1 signaling, RAS1, SUR1 signaling, CMP signaling, and NRG1. We also found no real alterations in the ED1 mutant regarding the response to ROS and other stressors. So while we know that ED1 is unlikely to be involved directly in any of the known farnesyl responsive pathways, unfortunately, we don't yet know what it does. However, there was another interesting observation which was related to farnesyl production. So we quantified farnesyl produced by the wild type or the mutant in supernatant and in association with fungal cells. And in the supernatant, within 24 hours, so a relatively short amount of time, we found 100-fold higher amount produced by the mutant compared to the wild type. And similarly, when we looked at membrane and intracellular fractions, we were not able to detect any farnesyl in the wild type, but we did detect it in the mutant. So in addition to being hypersensitive to farnesyl, the ED1 mutant is also producing more farnesyl. Interestingly, farnesyl has been shown by Han and co-workers to upregulate pathways which are involved in central carbon and energy metabolism. So this would link it maybe to the enhanced proliferation in vivo. And Navaratna and co-workers showed that if you add farnesyl exogenously to the infection model of systemic candidiasis in mice, you get much faster increase of kidney fungal burden, similar to what we observed when we compared the ED1 mutant to the wild type. So we decided, based on this, that it might be worthwhile to see what farnesyl is doing to growth of Candida albicans wild type in vitro. So this is ongoing work. What I show you has been reproducible and done often enough, but this is all ongoing. So if you take a condition where the ED1 mutant grows worse than the wild type and we add farnesyl to the ED1 mutant, we get no effect. If we do the same with the wild type, we see that the more farnesyl we add, the lower the OD gets. So the wild type has a tendency towards the ED1 phenotype in the presence of high amounts of farnesyl. If we do the same with citrate as a sole carbon source, where the mutant has an advantage, again, no effect of farnesyl on the ED1 mutant, but for the wild type, the more farnesyl, the higher the OD gets. So again, phenocopying to some extent the ED1 mutant. And you will see similar results if you do the same experiment on BSA. So although we don't really know what ED1 as a protein is doing, we know that deleting the gene leads to increased production of farnesyl and increased sensitivity to farnesyl. And if we expose the wild type to exogenous farnesyl while growing, we see that in those situations where we have a difference between wild type and ED1, the wild type is shifted into the direction of the growth phenotype of the ED1 mutant. However, if you would look at the absolute numbers, there is no complete phenocopy, and it might be interesting to see whether there is a difference in the effects of exogenous versus intracellularly produced farnesyl in Candida albicans. Also, we didn't see any effect of exogenous farnesyl on the ED1 mutant, which might indicate that the ED1 mutant produces so much farnesyl that the signaling pathways are in fact saturated. So this is something we hope to follow up in the future. And if anybody is working in that direction, we're happy to collaborate. Please let me know. Also, if you're interested in the strains. I'd like to finally point out that farnesyl not only affects Candida albicans, but also immune cells. So there's work by the Atkin, the Kotzer labs, and others. And it can also affect Candida albicans cell wall remodeling, which in turn might affect interaction with immune cells, which is something Rebecca Holt's lab has shown recently. So there's certainly more angles to be explored in the future. With that, I'm at the end. I would like to thank, of course, all my group and institute, but especially Christina, who will defend her PhD thesis soon and who did all the in vivo work with the ED1 mutant, Melanie, who's an alumni of my lab and did all the work on the farnesyl production hypersensitivity. I'm thankful to all the collaboration partners, especially Bernie, but also all the others who contributed to the work I presented today. And I don't have enough space on the slide to name all the other collaboration partners and other projects whom I'm really thankful to. All of this wouldn't be able without funding from various sources. And thank you very much for your attention. And now patiently will stand by until the question and answer session.
Thank you very much, Ilse. That's a really interesting project with some really interesting pathology. And I look forward to the discussion later. So as is our custom here on MycoTalks, we will hold questions until the end of the the talks by both our speakers. Can I please just remind you all, please to put your questions into the Q&A box, not into the chat box, and also preface your questions with the name of the speaker, so that Ilian and I can then disentangle all the various questions at the end of the session. Okay, so let's move on to the next speaker. And I'm really looking forward to this. This is my great friend and colleague, Al Brown, who is based with me here, as you can see, by our very formal backgrounds at the MRC Center for Medical Mycology at the University of Exeter, of which Al is a co-director. Now, I asked Al what he wanted me to say at his introduction, and in his typical modest fashion, he gave me 3 lines: PhD in 79, joined Aberdeen in 89, and then left and went to Exeter in 2019. Very modest, typically of Al, but I promised I would use this opportunity to internationally wind him up. And I thought what I would do in that case is to use a significant portion of his allocated time slot to go through and highlight in great detail all his many contributions to the field. Just kidding, obviously I'm not going to do that. It would take too long. So what I want to do, though, is just to highlight just 3 areas where he has made significant contributions to the field. That is in environmental adaptation, signaling and gene regulation in Candida, the regulation of Candida virulence and fitness attributes during colonization and disease progression, and the impact of fungal adaptation upon immune evasion. And in fact, his work in these areas has been recognized by several fellowships, including the fellowship from the American Academy of Microbiology, Royal Society of Biology, Royal Society of Edinburgh, and most recently, the European Confederation of Medical Mycology. So Al, you're going to talk to us today about fitness, memory, and immune evasion in Candida, and I hope I've wound you up enough, and I look forward to your talk. Thanks, Al.
Thank you, Gordon. So hopefully you can see my screen share now.
Al, we can see your presenter mode. We need to go to, there you go.
Is that better? Okay. So thank you very much, Gordon, for the wind up and for the generous introduction. Like Ilse, I am really enjoying these MycoTalks, and I've really enjoyed series 1 and very much enjoyed Ilse's talk. So thank you very much to the MycoTalks organizing committee for giving me the opportunity to tell this story about fitness, memory, and immune evasion in Candida albicans. So first of all, to remind us all why we're here, we're studying medical mycology because of the major impact of fungal pathogens upon human health, and this is beautifully illustrated by this picture, this figure from Felix Bongeman and David Denning's review showing the global burden of fungal disease and the annual incidence of many fungal infections, many of which have extremely high rates of mortality. And Candida species play large in this figure in terms of recurrent vaginitis, invasive candidiasis, and esophageal candidiasis. And the scene is set worse is made even worse by the global threat of emerging resistance to antifungal drugs that we all know about, the limited number of clinically used antifungal drugs. And I'm using this figure from Matt Fisher's review, this snapshot from 2017 showing the reported azole resistance, the emergence of azole resistance in fungal isolates country by country across the planet. So in Gordon's seminal review with many other colleagues, they highlighted 3 major priorities: the development of better and more effective antifungals, development of better diagnostics, and the need for a greater understanding of fungal biology and immunology that will help drive the development of these better antifungals and diagnostics. And my particular story, of course, lies in this area of hopefully contributing a little bit to our understanding of fungal biology and of Candida albicans in particular. Now, Ilse has already given you a nice introduction to Candida albicans. And so I just want to very quickly remind you it's an opportunistic pathogen, that many of us carry it as a commensal in the GI tract, oral cavity, or urogenital tract. that it can cause nasty mucosal infections like oral thrush and vaginitis, and in seriously immunocompromised patients, cause life-threatening systemic infections of blood and bloodstream and internal organs. Now, in the context of this particular talk, one of the things that really intrigues me is the ability of the fungus to thrive in really quite different niches, reflects its remarkable adaptability and its ability to, as I say, adapt to different microenvironments in the host. So what makes Candida albicans such an effective pathogen? And you've heard some of this from Ilse in the previous talk. It's not just, though, the ability to regulate a whole plethora of virulence factors. It's also the expression of robust fitness attributes that are very important for pathogenicity. So there's this battery of virulence factors, many of which Ilse has talked about. But in this talk, I want to focus on fitness attributes. And what I mean by that are these attributes that promote the physiological robustness of the fungus in different host niches. And these include its metabolic flexibility, its ability to adapt to different nutrient availabilities in different niches, and the ability to mount robust responses to host-imposed, or many at least, host-imposed stresses. Now, this adaptation, the adaptation of the fungus is really important, and I want to point out in this slide that it has to do so in the face of a complex, really complex host niches, where it faces multiple inputs that it has to deal with and adapt to. And furthermore, these inputs are dynamic in that they change over time. So each fungal cell is trying to perceive what's around it in the environment and adapt to these input signals. And as it does so and start to grow, it changes that in microenvironment. And the host, of course, will try and mount immune defenses against the fungus, and the fungus will try and respond and counteract these defenses. So these niches, the Candida albicans host interactions and host niches, are both complex and extremely dynamic. So I would argue that environmental adaptation is an important, very important aspect of fungal fitness in vivo, and hence pathogenicity. Now, a host of really great labs have investigated the signaling pathways that drive the responses of Candida albicans to specific stresses, nutrients, and developmental signals. And these pathways and their responses they trigger promote fitness in vivo, the survival and potentially the growth of the fungus, potentially ultimately leading to colonization and infection. But because of the way we study these pathways and dissect these pathways, we're often guilty of considering these pathways in isolation, when actually, in reality, these pathways are, there's multiple forms of crosstalk between these pathways. And so these pathways are interconnected within a signaling network. Furthermore, something I'd like you to consider is that some specific environmental inputs in certain niches are temporally linked, in that one type of input is sometimes or semi-frequently followed by a second type of input. And in these circumstances, this creates a selective pressure for the creation, loss, or tuning of these connections, the crosstalk between pathways within the signaling network. Now, there's a fantastic paper, I think an inspirational paper. It was published some years ago by Yitzhak Pilpul's group that has really influenced my thinking, at least over the last decade or so. This is a paper about adaptive prediction of environmental changes by microorganisms. The way I see it, there's two key things that Yitzhak Pilpul is saying in this paper. The first is that some microenvironments that microbes occupy are reasonably predictable, as I've said, in that one type of input is often followed by a second specific type of input. And he used in the paper examples of the sequential usage of carbon sources by E. coli in the gut, and also uses as an example sequential stresses faced by Saccharomyces cerevisiae during wine fermentations. So some microenvironments are reasonably predictable. And if microbes occupy such environments, then they will gain a fitness advantage if they evolve anticipatory responses that prepare the microbe for the second input when it detects the first input, but even before. So it's activating response one, not just activating the response to the initial input, but also activating a response to the second input before it even occurs. So they'll gain a selective advantage by doing that, by activating an anticipatory protective response. And so Yitzhak Pilpul argues about that there's an evolution of networks, and some of the evolution of these signaling networks may drive the emergence of anticipatory responses. And this phenomenon of adaptive prediction seems like a form of fungal memory that reflects, I would argue, relatively recent evolutionary pressures that they faced in their niche. So does adaptive prediction occur in Candida albicans? Now I'm going to describe some examples of adaptive prediction that we've been studying in my lab, but I should point out just before I do this, that Berne Hubi and Sascha Brunke have also described examples of adaptive prediction where hyphal development is linked to, for example, micronutrient assimilation and Candida lysine production. But the first example that we found and we've studied is a phenomenon called glucose-enhanced oxidative stress resistance. And what we find is that exposure of Candida albicans cells to glucose protects them against subsequent oxidative stress. And this was particularly intriguing to me because the exact opposite happens in Saccharomyces cerevisiae. Growth in glucose in Saccharomyces renders Saccharomyces more sensitive to oxidative and other stresses. So the first evidence for this was when we looked at cells growing on lactate, and in control cells, they stayed on lactate. Other cells were exposed to glucose or even 20% plasma. And what we found is that these cells that were exposed to glucose or plasma were much more resistant. They retained their viability to a much greater extent than the control cells when exposed to an acute dose of hydrogen peroxide. But significantly, glucose exposure also protects against subsequent attack by human neutrophils. And what we've got is a barcode sequencing project where differentially barcoded but isogenic Candida albicans strains are pre-adapted in different ways. The controls with 0% glucose, or the experimental cells exposed to 1% glucose or 1% plasma. They're then exposed to neutrophils. And the viable cells, you can see an enrichment of viable cells following neutrophil exposure. For those that are exposed, pre-adapted by seeing plasma or glucose compared to the control cells. In the absence of neutrophils, there was no difference. So there's protection against subsequent attack by neutrophils. And that also, pre-adaptation to glucose also enhances fitness in vivo during systemic infection. And what we've got here is another barcode sequencing experiment with differentially barcoded strains pre-adapted in different ways, and then injected in the tail vein of a mouse. And then a couple of days later, we look at the fungal burden in the kidney, and we can see enrichment of cells being exposed to glucose or plasma compared to untreated glucose-grown cells. Sorry, untreated cells that are purely grown on lactate in the absence of glucose. So with glucose-enhanced oxidative stress resistance, the first input is, of course, glucose exposure. And Candida cells don't just tune their metabolism to the glucose availability, they also induce oxidative stress genes, which protects the cells against a subsequent oxidative stress that they haven't even seen yet. So when might this be of relevance in a human context? Well, cells growing in the gut, for example, are not, the colon, for example, are not seeing glucose. But if they're exposed to blood in any way, then they'll detect the glucose. They'll increase their resistance to oxidative stress. And this might protect them against subsequent attack by the neutrophils that are circulating in the blood. So we argue that this is an anticipatory response whereby exposure to glucose provides protection against subsequent neutrophil attack. Another example, we've discovered another example of adaptive prediction in Candida albicans more recently, and this is lactate-induced beta-glucan masking. Now, in this phenomenon initially this whole story started with Jules Enna when she was a PhD student in my lab some time ago. She's now has her own position at the Pasteur. And she discovered that exposing Candida cells to lactate resulted in dramatic changes in cell wall related phenotypes, including recognition and immune responses. Liz Ballou extended this story by studying beta-glucan exposure on Candida cells that were exposed to lactate compared to glucose-only grown cells, and found that cells that were exposed to lactate for 5 hours exposed much less beta-glucan compared to the control cells. I should say this is flow cytometry experiment looking at 10,000 populations of about 10,000 cells that have been stained with dectin-1 to highlight exposed beta-glucan on the cell surface. And this beta-glucan masking by exposure to lactate has been confirmed by quantitative fluorescence microscopy by Jude Bain, looking at the exposure of beta-glucan on the cell surface by dectin-1 staining. And here's the control cells just grown in glucose. And here are cells that have been exposed to lactate for a period. And you can see much less staining and a much lower fluorescence score. So exposure to lactate induces beta-glucan masking. Similarly, exposure to hypoxia induces beta-glucan masking. Compared to normoxic cells, there's considerable cell wall remodeling when exposed to hypoxia. If you can see the green here, in the normoxic cells, there's more beta-glucan exposed on normoxic cells compared to hypoxic cells by fluorescence microscopy. And again, as Arnab showed, by flow cytometry, there is much less beta-glucan exposed on hypoxic cells compared to normoxic control cells. And again, iron depletion induces beta-glucan masking, induces significant cell wall remodeling, iron replete cells, iron depleted cells. Here, the exposure of beta-glucan on the cell surface is highlighted in yellow. And again, this is by dectin-1 staining. These are iron replete cells, much less beta-glucan exposed on iron depleted cells. And this microscopy confirmed by examination of large populations of cells by flow cytometry, again by Arnab. The control iron replete cells, the experimental, and here are the iron depleted cells, much less exposure of beta-glucan on the cell surface. So fine, we have a number of specific signals, iron depletion, hypoxia, and lactate, that trigger beta-glucan masking by some means through cell wall remodeling. So what is going on here? How is beta-glucan masked? Excuse me. Well, it's well known that the cell wall, the Candida albicans cell wall, contains a lot of beta-glucan, but this beta-glucan in this lovely scalar model in Megan Lenardon's paper, these green beads here, this beta-glucan is almost all in the inner cell wall, and is masked by these beautiful Manan fibrils in the outer cell wall, these Manan fibrils here, masking the beta-glucan, hiding it from, if you like, the immune system. So most beta-glucans in the inner cell wall, buried beneath the Manan outer layer. But a number of groups, and these include Rob Wheeler's group, Jesus Pla, Fernandez Ruiz, Becky Hall with Neil Gao, Todd Reynolds, Aaron Newman, have shown that genetic or pharmacological perturbation of the cell wall, and particularly the Manan layer of the cell wall, leads to beta-glucan exposure. So the outer layer of Manan masks the beta-glucan in the inner layer. But we still see exposure of some beta-glucan on the cell surface, despite this Manan masking. And this beta-glucan is exposed at bud scars on the cell surface, but also in punctate spots on the lateral cell wall of the cell. And Leandro Assis has done some high resolution fluorescence microscopy and imaging of these punctate spots. These punctates, some of these punctate spots represent what look like pores of glucan leaching out through the Manan outer layer. So how does this masking occur? This exposed beta-glucan, how is it masked? Well, Delma Childers has looked at the secretome, has looked at which proteins are upregulated in the secretome of Candida albicans cells that are exposed to lactate and also hypoxia, two of these masking-inducing conditions. And there are 4 proteins that were induced in common in response to these conditions. And the most highly upregulated of these is the XOG1, which is major exoglucanase. Also on this list, another protein on this list is ENG1, which is an endoglucanase. Delma followed this up by making an XOG1 knockout and showing that XOG1 knockouts show attenuated masking, a lower degree of masking compared to wild type cells. So also Helping Lu's group has shown that similarly inactivation of ENG1, the endoglucanase, also attenuates lactate-induced beta-glucan masking that can be seen in wild type cells. Also, you get an increased exposure of beta-glucan on the cell surface in ENG1 cells. So there are 2 main mechanisms by which beta-glucan appears to be masked. The first is by the main Manan outer layer, but the second mechanism appears to be glucanase shaving by the exo and endoglucanases, the shaving of exposed beta-glucan on the cell surface by these glucanases. Okay, so how is beta-glucan shaving activated then? So a combination of work from Liz Ballou, Gabriela Avalar, and Arnab Pradhan has led to the following summaries, if you like. And the first is that lactate-induced beta-glucan masking is dependent on the G protein coupled receptor GPR1 and its partner protein GPA2, a G protein alpha subunit. Inactivating these proteins blocks lactate-induced beta-glucan masking. Should highlight that the GPR1 and GPA2 have been studied by Patrick Van Dyke in the context of morphogenetic regulation. But GPR1 is actually the closest homologue in Candida albicans to mammalian GPR8, GPR81, a lactate receptor. So as I've said, lactate-induced masking is dependent on GPR1 and GPA2. In contrast, hypoxia-induced beta-glucan masking is dependent on the generation of hydrogen peroxide in the mitochondrion, in other words, mitochondrial ROS signaling. And I should add that hypoxia-induced beta-glucan masking is not dependent on GPR1 or GPA2. Meanwhile, the beta-glucan masking that's induced by iron depletion is dependent on the FTR1 iron transporter and receptor, and also the CEF1 transcription factor that's been characterized by Suzanne Noble's lab and is very important for mediating iron responses, or responses to iron depletion, I should say. So we have 3 different upstream pathways mediating responses, or the beta-glucan shaving in response to iron depletion, hypoxia, and lactate. But all 3 of these pathways feed into protein kinase A signaling pathway, the site at different levels into the cyclic AMP, adenyl cyclic AMP protein kinase A signaling. So what appears to have happened during the evolution of signaling networks in Candida albicans, at least, is that there appears to have been recruitment of evolutionarily conserved input-specific signaling pathways, upstream signaling pathways, that then feed into, well, another evolutionarily conserved signaling pathway, the protein kinase A signaling pathway, to trigger beta-glucan masking, shaving, and masking. So why am I going on and on about beta-glucan masking? Well, it's because we think it's highly relevant and strongly influences immune recognition. So beta-glucan masking correlates with attenuated phagocytosis by macrophages. And this is the case for cells that have been exposed to lactate. Lactate-exposed cells show decreased macrophage phagocytosis, as do cells that are exposed to hypoxia compared to normoxic control cells, as do cells that have been subjected to iron depletion compared to iron replete cells. So beta-glucan masking correlates strongly with attenuated macrophage phagocytosis. And it also correlates with attenuated cytokine responses by human PBMCs. And what this graph shows is work by Gabriela Avelar. And she looked at about 39 different conditions. She allowed Candida albicans to adapt to 39 different growth conditions and looked at the effect of these different growth conditions on beta-glucan exposure. And some of them, compared to the control cells, led to increased beta-glucan exposure, some to decreased beta-glucan exposure, i.e. beta-glucan masking / shaving. And the extraordinary thing for me about this figure is that when she measured TNF-alpha production, the stimulation of TNF-alpha production by human PBMCs, there's a very, very strong correlation between, and highly significant correlation between the levels of beta-glucan exposure and TNF-alpha production by the PBMCs. Furthermore, beta-glucan masking appears to correlate with enhanced fitness in vivo. Now, a few years ago, Pavelka's group published that there's increased beta-glucan exposure on Candida albicans cells correlates with reduced gut colonization. And the way they showed this was to examine a number of Candida albicans cell wall mutants that displayed increased levels of beta-glucan on their cell surface. And they demonstrated that correlated with reduced fungal burdens in fecal samples. Now, more recently, Gabriela Avelar, with help from Ivy and Rife, have looked directly at Candida albicans cells in the mouse gut, and in particular in specific compartments of the mouse gut. And they did this by imaging flow cytometry of the Candida albicans cells, isolated specifically from each of these compartments, isolated directly from these compartments. So the first point from this first graph is where we're looking at beta-glucan exposure of cells, let's say wild-type cells from these different compartments. And here we have the small intestine, the cecum, and the large intestine, the colon. We can see that cells in these different compartments are exposing different levels of beta-glucan. But one of the really interesting things for me is that we also, when Gabriela looked at the GPR1, GPA2 double mutant, and remember this mutant is defective in lactate-induced beta-glucan masking, she saw a significant difference, a significant increase in beta-glucan exposure for cells that lacked the ability to mask beta-glucan in response to lactate. This wasn't the case in the cecum and the small intestine, but it was the case in the large intestine in the colon. So cells that are unable to mask in response to lactate expose more beta-glucan in the colon. And furthermore, this correlated with a significant reduction in the fungal burden in the colon. So blocking GPR1, GPA2 signaling increases beta-glucan exposure in the colon, and we see a corresponding reduction in the colon in Candida albicans colonization in the colon too. So beta-glucan masking appears to correlate with enhanced fitness in vivo. I should add, of course, I should say that there's an awful lot going on in the colon, not just the presence or absence of lactate and other short-chain fatty acids. So what I'm trying to say is that lactate-induced beta-glucan masking might contribute to the ability, along with a number of other factors, to the ability of Candida albicans to colonize the colon efficiently. So to start summarizing, beta-glucan shaving, we argue, is an anticipatory response that promotes immune evasion and helps to protect Candida albicans cells from phagocytic attack. Now, lactate produced by host metabolism or the metabolism of the microbiome doesn't just induce a metabolic response. It also induces beta-glucan masking, which reduces phagocytic recognition and thereby protects cells from phagocytic killing. Similarly, hypoxia induced by a combination of host and fungal metabolism doesn't just induce a metabolic and hypoxic response, it also activates beta-glucan masking to protect, to attenuate phagocytic recognition. And host nutritional immunity leads to iron depletion around foci of Candida cells during infection. And this doesn't just trigger iron assimilation, it also triggers beta-glucan masking again. So all three of these host inputs lead to an anticipatory response, beta-glucan masking, that protects from subsequent phagocytic attack. Now, even if some cells are recognized, there's another form of anticipatory response, the glucose-enhanced oxidative stress resistance, whereby glucose exposure doesn't just increase or tune metabolism, it also increases resistance to stress and hence phagocytic killing. I'm going to skip this slide, I'm afraid, in the interests of time, and summarize some main conclusions. So what I've tried to tell you is that predictable changes in the microenvironment can lead to the evolution of protective anticipatory responses. And this appears to have happened in Candida albicans, in that it's developed protective, it's evolved protective anticipatory responses that protect against immune responses. And these enhance the fitness of Candida albicans in vivo. And I would argue that they probably represent a memory of relatively recent evolutionary pressures that the fungus has faced. If I'm allowed just a few seconds to thank a brilliant team of people who worked on this project when I was in Aberdeen, and also a great team who've continued this work when I moved down to Exeter. I'd also like to thank my colleagues in the MRC Centre for Medical Mycology, brilliant set of supportive colleagues. Neil Gow in particular, Lars and Mihai are collaborators on this particular work that I've talked about. Gordon's been enormously helpful. Liz has now, who worked on this particular project, has now joined the team as a PI. I'd also like to thank long-term collaborators in Aberdeen and Newcastle, Janet Quinn, and also Christoph and Bernie, who've been fantastic collaborators, and the generous support from my funding bodies and Leonardo for the artwork. And thank you very much for your attention.
Thank you, Al. That was a really very fantastic presentation. So as we will do in the past, what we will do is we will start a tag team between Ilse and Al in terms of questions. Can I just please ask all attendees, just remember to put your questions into the Q&A box, and please preface that with the name of the speaker. That will allow Ilian and myself to be able to distinguish the questions quite quickly as we move forward. And I'll hand over to Ilian to start the questions for Ilse.
Right, fantastic talks, first of all. That was amazing. So first I will start with a question for Ilse. So what is the function of EED1 in Candida, and what do we know from other Candida species, such as, for example, Candida glabrata? Or have you tried to express EED1 in Saccharomyces cerevisiae to look further for the mechanism?
Okay, let's start with the question whether or not there are ED1 homologs in our Candida species. Geraldine Butler once pointed out that there is a similar gene in Dubliniensis. And we also played around when Peter Stipe was still in the mycology field and tried, I think, deleting the more or less homologue Indobliniensis and tried to put in the Albicans ED1. However, that mutant was extremely unstable. This is something we've seen also when we tried to clone the ED1 gene into E. coli. There's a lot of recombination events, weird things going on. If you try to express it in E. coli, they either die or chunk out the plasmid. We tried expression in Pichia and Saccharomyces, and it didn't work. And this wasn't done by us, but by labs who know how to do it. So this has been a real disadvantage because we were not able to develop the tools necessary to really molecularly dissect the function of the protein. So we got stuck there at some point, not for lack of trying, unfortunately. This also means that ED1 might be something relatively specific to albicans within Candida. And this might have to do with the fact that the other Candida species do not filament as much, even Dubliniensis, under most conditions. But we haven't followed up on the question of ED1 in other Candida species. Did that answer all aspects?
Yeah. I think it opens actually very interesting talks here as well.
You want me to take over, Ilya? Okay. So, several questions and not many of them that I can group, unfortunately, which makes it difficult. So first question for you, how long lasting are these adaptive responses in the cells? How quickly do they restore to normal?
Okay, that depends. It depends on the nature of the input. If you take away the signal, so for example, lactate-induced beta-glucan mask, if you take away the signal, it takes some hours for the signal for, let's say, exposure to normal levels of exposure to return. With hypoxia, it could take, the changes are faster. If you maintain the signal, so if you maintain lactate and keep subculturing the cells, so they're not going into stationary phase, they're still growing, then the masking can be retained for quite a few days. Okay, so it depends on I'm not quite sure what the person's really asking. So if you remove the signal, then after some hours it can disappear. If you retain the signal, then the masking can last for a long time.
Okay,
does that answer the question?
I think so. Yeah, I mean, I think it was just a question of how long, if you stem this away. But then I guess a related question about the kinetics, which is what you were going on about.
Right.
So could this these conditions basically just differ the kinetics of cell wall deposition, and could that really be what you're seeing is actually a metabolic change as opposed to an adaptive response as you were postulating?
Yes and no. I don't think it's simply to do with metabolism, but it is related to growth. So first of all, the masking is not about a sudden change in growth rate. The masking, that's not what's explaining the masking. For example, the shaving phenomenon is dependent on secreted glucanases that come around and shave off the enzyme. But I think the exposure of beta-glucan is, of course, related to growth, because a lot of the exposure is occurring at bud scars on mother cells. So the more growth there is, the more division there is, there'll be more exposure. And I can't help feeling, I don't know what the origin of the punctate spots is. I've got some random thoughts, but I don't know what the origins are. But I think growth does influence the rate at which they may emerge too. So I think the growth, the synthesis of new cell wall, the generation of bud scars, it does underlie the exposure. Growth phase does influence the degree of masking. Neil Gow's team has looked at the degree of masking during growth phase. And if you look in stationary phase, cells there are exposing much less beta-glucan than cells in exponential phase. But I think that's a balance of the kinetics of growth and the emergence of beta-glucan at the surface and the masking of the beta-glucan. So I don't know if I got out of that one.
Excellent. I'll hand you back to Ilya now.
All right, so question for Ilse. So I'm grouping a few questions here, but the point is, so people are asking what is the EED function during clinical disease? And for example, how do kidney intrinsic damage responses and markers such as BUN, bradykinins and others change in the face of lethal infection with the wild type versus the mutant?
Yeah, so we've measured blood urea nitrogen, creatinine, and KIM1 as a tubular damage marker. That's actually in the paper, and there's no difference between mice infected with the wild type or the ED1 mutant. The curves are literally superimposed, which surprised us. We actually expected to find differences, but we didn't.
So related to that question, so if you go with yeast lock versus Hyphy sufficient strain of Candida, not with that pathway, yeah, is there any difference into that?
So we've actually run, and that's all in the supplementary data, and I didn't mention it today, we used the NRG1 strain from Stephen Sewell also as a control to make sure that our doxycycline system works. Now that strain was strongly attenuated. You see higher fungal burden with the NRG1 yeast than with the filamentous version or the wild type, but they never reach quite the levels of the ED1 mutant. So it really, when it comes to pathogenicity by yeast cells, quantity might really be important. There are a few interesting older publications, one from Frank Arts and Donna McCallum, for example, where they did a meta-analysis of a large array of mouse experiments using different inocular sizes and strains. And interestingly, I mean, these were all filamentous strains. Usually when the mice become moribund at the end point, you always reach a similar fungal burden in the kidney. And in another study where different wild type strains were compared with differences in filament formation, quantitative differences, not black and white, and the infectious inoculum was adjusted to get to the same mean survival time. Again, at the end point, what you found were significantly more yeast cells or fungal burden in those strains with less filamentation. So, you know, if you use the approach comparing two strains using the same inoculum or same infection dose, you simply, you know, because you're in the balance of the cells being killed off, probably especially in the early phases, you might never reach the level which is detrimental to organ function. And this, I think, is also interesting if you think about Candida glabrata in a clinical setting in human patients. In mice, even if you put in a large and infectious dose, usually kidney burden never get as high as we see with the ED1 mutant, and the mice do just fine. Maybe there is something different in human patients which allows more proliferation.
Thank you.
So I'm going to combine a couple of questions. They're roughly linked here. So first of all, is the effect on masking, unmasking additive between the different stresses? Are there variations at the single cell level between different yeast cells, and can the yeast cells themselves influence adjacent yeast cells in these responses?
Wow.
You can break it down if you like.
Okay. There is definitely, I mean, you could see from the cytometry curves that I showed, there is definitely population heterogeneity. Some cells are masking more than others. And this isn't simply about mother-daughters or, you know, new cells and old cells and budding. There's more to it than that. I have pet theories about this, that this relates, and this is why I mentioned the signaling networks and the importance of signaling networks. And I think stochastic differences between cells will lead to different amounts of exposure on the cell surface and potentially a different amount of masking. But there is definitely a high degree of population heterogeneity. And there's strain to strain variation as well, I should add. Second part of the question, can adjacent cells influence masking or beta-glucan exposure? That's a cracking question. Of course, we've been using planktonic cultures, and so or scraping cells from the large intestine and looking and seeing the population heterogeneity of them. But so we would be breaking up adjacent cells. We've not looked at, we've not done the right experiments to test whether an adjacent cell can affect exposure in another. But given that secreted glucanases are involved, then potentially yes. That's the answer.
Okay, and then the last part of that question, which was the additive nature of the different stresses on masking versus unmasking.
Another cracking question. So I think I got my excuses in early about the complexity of the colon and the number of signals that will be in the colon. And of course, it's not just lactate in there. There's lots of other short chain fatty acids in there, butyrate, propionate, and they actually influence masking as well. Acetate leads to beta-glucan exposure, not masking. And butyrate leads to exposure, not masking. And so you've got this complex mixture of short chain fatty acids. So we have looked at the combinatorial effects of these, and I can't remember the result, but I can remember that if you look at the hypoxic signal versus the lactic signal, for example, hypoxia is like a dominant, a really strong masking phenotype. But the nub of this question is, the combinatorial effects of different inputs upon masking, and that must be really, really complex. And that's why we're trying to look at what's happening to cells in vivo in these compartments. Even if we don't understand all of the signals that are affecting it, at least I think we've been able to show that the GPR1, GPR2 signaling pathway is important, what contributes rather to colonization.
Okay. Thanks, Al. Back to you, Ilya.
Thanks. And just to add, actually, to Al's thoughts here, there is also a microbiogeography, which is basically horizontal. So same colon, it's like 2 different continents. So there is a lot of that there. It's a very fascinating area. So, there is, Ilse, there are a few questions on the farnesol. I'm combining them. So farnesol is a byproduct of the sterol pathway. How do EDT mutants respond to azos or other ergosterol synthesis inhibitors? And then how long is the effect of the farnesol? So if you take cells out of the organ, is that actually, or when there is actually no farnesol stimulation anymore, do cells recover?
So to the first question, Ronnie Martin, when he was a postdoc with Bernard Huber, actually he published a paper on ED1 and how its function is related to UME6, for example. And he did transcription profiling. So if you look into that, there are a few genes within the ergosterol biosynthesis pathway up and down regulated, but without a very clear picture. We tested for antifungal sensitivity. The ED1 mutant is slightly more resistant, but if you use cell wall disturbing agents like Congo red, calcifluor white, it's more sensitive. There certainly is something going on. We see a little bit more menin and chitin exposure and slightly less beta-glucan exposure. So I would, there certainly is something different in the surface. The other question is, so what would happen if you take the farnesol away? So what we have done is experiments where we let the ED1 mutant grow under Hyphy-inducing conditions for 24 hours. So you end up with a more or less pure yeast culture. We wash these yeast cells and then put them into Hyphy-inducing conditions. So that should have removed the majority of unbound farnesol. And we like to call them secondary yeast cells. They do much less germ tube formation. They're more sensitive even to exogenously added farnesol. So it seems that there might have been, by whatever mechanism, some kind of priming of those yeast cells to be even less likely to filament in the context of farnesol. So I can't answer how long the effect lasts, and it might depend on what type of effect you look in terms of farnesol. Another thing which is peculiar with the ED1 mutant, even though it's hypersensitive to the filament inhibiting effect of exogenous farnesol, it's more resistant to the toxic effect of farnesyl. So if you expose wild-type cells to very high concentrations, you see cells dying off. And that doesn't happen with the ED1 mutant. So it's not simply more production, and maybe therefore you need less exogenous because there's so much farnesyl already inside. It's also that there is something different in those yeast cells because they're not so susceptible to toxic farnesyl effects.
And are they more susceptible to azoles, to fluconazole, for example?
They're slightly more resistant.
They're more resistant, okay.
More resistant for whatever reason. He looks as confused as I am by that, yeah. Yeah,
so Gordon.
Okay. Oh, question about that pore that you were showing. So there's interest on that. Comment from Mark Resnick about this. So when you see stain Hyphy for beta-glucan, you see similar punctate structures. Is this the same thing? And do you know if it's a true pore and possibly what its function is? Mark was postulating that maybe it's to facilitate the efficiency of underlying transmembrane transporters, or something along that lines.
I showed the pore because I'm fascinated. I showed that structure because I'm fascinated by it. I think to truly understand the masking phenotypes, we need to understand what's the nature of the exposure. So this pore-like structure, something actually like a positive function. I hadn't really considered that, Mark. I was, I'll tell you again, another pet theory, and this might relate to growth. I'm wondering about, what happens when an extracellular vesicle transits the wall, pushes up through the inner layer, and then emerges through the outer Manan layer. Does it drag? I'm wondering if these pore-like structures just represent beta-glucan that's being dragged through the wall by an extracellular vesicle as it leaves, crosses the wall and emerges. That's my pet theory. We haven't pursued that. If anyone has the wherewithal to block or regulate extracellular vesicle production, I would love to hear about that and maybe collaborate and see if that is the basis for these punctate structures in the lateral cell wall. But that's an interesting idea, Mark. I hadn't considered actually something active and positive like encouraging transport across the wall. I hadn't considered that.
Thanks. So back to you, Ilya.
So there are a couple of questions here on basically if you have used RNA-seq or proteomics to look basically of how mutants versus how the mutant versus the wild type would look in the kidneys once you isolate them, and to come up with, to look at pathways that might be affected. And the other related question is if in the context of farnesyl, you have looked by proteomics, similar idea.
Yeah, so we haven't done RNA-seq from kidney samples. It's on the list. It certainly would be interesting to do, and especially for the early time points, fungal burdens should be sufficient to get meaningful data. For the proteomics analysis, so this was also on the list when we submitted the paper. Then one of the reviewers came back and asked for transcription profiling. And the reason we didn't do the proteomics and include it is simply that to really get a complete picture, I would like to do it under different conditions where either wild type or mutant has an advantage. And then if you do secretome and cell-associated proteome, and do it over a time course, you simply end up with something which takes a while. So we didn't do it yet simply for time reasons. But it's an obvious thing to do, especially when it comes to metabolism, because as I've learned from Al, a lot of metabolic genes are regulated post-translationally.
Thank you, Ilse. So to Gordon.
Okay, so Al, I'm going to try and tie two questions together, which are going to be again, very diverse questions, and see what you do with that. So other than macrophages, have you looked at other immune cells and do they respond? Does the Candida masking respond in the same way in terms of things like uptake by neutrophils, for example? And then another question, so the program that you get in response to different mammalian host niches, right? The different cells that are there. do you think that Candida has specific programs to respond to each of those tissues? And could that be a readout of the tissue in which they're located?
Okay. First question, I'm afraid despite your influence, Gordon, we've only just looked at macrophages, some different types of macrophages, but usually bone marrow derived macrophages, murine ones, and then looked at cytokine responses from human PBMCs. So that's, I'm afraid that's all we've done. We've not looked at other cell types. Second question, programs. I mean, Candida albicans is smart, but it's not that smart. I think the cells are just trying to detect what's immediately around it in the microenvironment and respond to them. So I guess that means if tissues and niches are different, there may be, an overall transcript profiling of different tissues. You may see subtly different responses that may be markers for particular infections. Is that what they're really going for? Diagnostic markers of where the infection is? But I don't think Candida is that smart.
I don't think they were asking diagnostic markers, but more about whether they...
I'm overreaching, am I? Okay.
But anyway, that was the question. Back to you, Ilian.
So there are a lot more questions, but we are almost running out of time. So I'll close with a question to Ilse, which is basically a bridge between Ilse and Al. So what is the cell wall structure of the EED1 mutant?
Yeah, so in collaboration with Neil Go, we looked at cell wall composition. There were no real striking differences. If you look at exposure of Menan, so using flow cytometry, Al, we listened to your talks to learn how to do it. We see more Menan and chitin being exposed and less beta-glucan. If you look at cells, yeast cells of the wild type and the ED1 mutant grown in normal YPD with high glucose overnight. If you look at what I mentioned, the secondary yeast cells produced when under filament-inducing conditions, the mutant switches back. Or if you do the same with the wild type at a very high inoculum dose, so to force the wild type to get to the quorum sensing benchmark and produce these cells. Then for ED1, these differences compared to the primary wild type strains become more pronounced. For the wild type, for the wild type, then in the secondary yeast cells, chitin also goes up, but Menan and beta-glucan are the opposite compared to ED1. So you get also more beta-glucan and less Menan. But we didn't test that extensively with different nutrient sources. As we know from Al, this all might have an impact as well. But yeah, there are differences.
All right, thanks to the speakers for the wonderful talks. Thanks to everyone participating with us today. So Gordon, do you have anything to add? And we'll be closing the session. It was great to see everybody.
I'll just take your thoughts. Thanks to the speakers for their really great talks, and thanks to the attendees and the participation. We'll see you next month.
Yes. Bye, everyone.
Thank you very much, everyone.